Hi, Professor. So for my written assignment, I decided to do the transcription and translation. So to begin, what is transcription? Transcription refers to the production of RNA using the information in the DNA. So basically, when transcribing a strand, we are rewriting it from DNA to RNA using the nucleotides along the DNA strand. And the bases of that are the A, G, C, or T. And so for RNA, the bases are A, G, C, or U. And so when transcribing, it's important to remember that A equals U, T equals A, G equals C, and C equals G. And so then transcribing makes a messenger RNA or an mRNA. So to continue, translation is the synthesis of a polypeptide using the information from the mRNA. And so when we finish transcribing it there from the original DNA. During this stage, there is a sort of change in language. It's where the cell translates the nucleotide sequence from the mRNA into an amino acid sequence of a polypeptide. And this whole thing takes place in the ribosome. And that kind of change of language is using the codon chart. So after translating, you kind of line up and then it's in groups of 3, and that's the codon, and that's the kind of change in language. And so then, I'm not very good at using this recording, I'm so sorry. So then using what we did for the assignment, we are first going to recognize the 5 or 3 caps, so it's going to be the ends of the codons. And so that's the untranslated region on the ends of the coding segments. Then we have the promoter, which is used to establish where the RNA synthesis begins. And then we recognize the start codon, which is the AUG codon. And that's the codon that gets transcribed first. Then we have the stop codon, which it kind of marks the end of the translation. In this particular strand, the stop codon was UAG. And yeah, sorry if there was so many technical difficulties. I'm not very good at using Screencast-O-Matic. Thank you. There you go.